Hello, everybody. Hi, I'm Veronica Hall from Claramedics Biosciences, and I'm here to present about biofilms and respiratory infections and kind of a different approach of how to address bacterial biofilms. Now, if I can actually get this and go the wrong direction. It's really the challenge. About 80% of all bacterial infections are biofilm driven, but we're still addressing just the bugs themselves, but we're not addressing the bacterial biofilm. Thank you. So really, success requires different approaches to address the biofilm. And what we're looking at is we're looking at an approach that enhances frontline therapy, is universal across biofilms, has broad indication potential, and also addresses AMR. We currently have two products in our pipeline. Our lead product we call CMTX-101, which is an anti-biofilm antibody. It's therapeutics, clinical stage. We also have CMTX-301, which is an elite optimization, and it's a preventative mechanism for antibiofilms. So really, we're kind of getting back into what the challenge of a biofilm. As I mentioned earlier, 80% of all bacterial infections are associated with biofilms. This leads to about $50 billion in the US healthcare and cost, and also is primarily responsible for approximately 20 to 40% failure of frontline antibiotics. For those who aren't familiar with biofilms, they're a community of bacteria that are surrounded by an extrapolymeric substance that protects and shields the bacteria from host immune effectors and from antibiotics. Okay. So most people think of biofilms as chronic versus acute infection. I'm here to say that biofilms are with all infections, and there's some recent data that just came out of I think it was written by first author Colpin out of Thorax, was showing that the biofilm in sputum samples of community-acquired pneumonia patients. They also have biofilms are well documented in COPD and in CF patients. There's also now documentation of biofilms in NTM lung infection. So biofilms are acute and chronic, kind of a different way to think about it. And we're here to try to address the biofilm problem. So what is our approach? As I mentioned, the extrapolymeric substance, biofilms differ across bacterial species. So how do we do this in universal target? There is one universal component of biofilms, and that is an extracellular DNA lattice. That is the structural component, and it is held together at the vertices of the DNA strands by the protein family DNAB2 protein families. These DNAB2 proteins are highly conserved where they bind DNA across bacterial species. And there is no mammalian homolog. So what we do is our technology targets DNAB2 proteins, and they rapidly collapse these bacteria biofilms. How do we do this? So in the biofilm, in the microenvironment of the biofilm, the DNAB2 proteins exist as free and biofilm-bound proteins. Our antibodies come in, they actually bind to the free. So they don't have to touch the biofilm. They don't have to penetrate the biofilm. They just have to get close enough to bind the free DNAB2 proteins. By doing this, it disrupts the equilibrium. Once the equilibrium is disrupted, the biofilm starts releasing DNAB2 proteins. And it's basically, if you've ever played a game of Jenga, eventually you pull enough of those blocks out, it all comes crumbling down. When it comes crumbling down, this enhances immune clearance. We have pictures. You've got your neutrophils just sitting at the top of these biofilms, waiting to attack. You also allow antibiotics to get in and clear more effectively. And also, although the least study of the mechanisms, it shows reduced site-specific inflammation. One of the other advantages of this is that we don't have a one-for-one. We only have to remove about 1/3 of the DNAB2s from the biofilm to cause collapse. And I mentioned that it is a fairly universal target. As you can see here, we have shown in vitro activity against all of your key respiratory pathogens, your escapee pathogens, your NTMs, in vitro and starting to generate in vivo data. We've also showed that in most cases, we restore these pathogen sensitivity to planktonic levels against the major classes of antibiotics, and that's what's shown in here. So we are pathogen agnostic, we are antibiotic agnostic, and can I give you an example? I couldn't show this data, but we took an isolate from Pseudomonas isolate from a CF patient that had an MIC, I believe over 4,000 micrograms per mil, grew it up in a biofilm, treated it with CMTX 101, and we reduced its, became more sensitive to tobramycin. I believe it became about 512 micrograms per mil, which is now a clinically achievable level of antibiotics. We have a tested CMTX 101 in a lethal an LD80 model of Pseudomonas challenge in C57 black 6 mice. In this model, to kind of give you a little bit more, mice are challenged with LD80 Pseudomonas. 24 hours later, we treat intravenously with 101 at various dose levels, and we have seen increased survival with CMTX 101 treatment to about 80%. We actually see our highest survival with our 5 mg per kg dose. You can see why didn't 30 mg, why didn't it plateau? We speculate that there's a potential prozone-like effect, which is well documented in diagnostics with antibodies and is currently starting to get more documented in the in vivo setting with antibodies. And now we are starting, as I mentioned, we're in the clinical phase. We just completed dosing in our healthy volunteer portion. I actually think we're cleaning almost all of our sample collection as well in our healthy volunteers, but we had a single ascending dose healthy volunteer study. There's been no reported SAEs. Each escalation was reviewed by Safety Monitoring Committee, as well as progression to our phase 1B study, where no safety concerns were identified. We are currently enrolling in our phase 1B study. This is going to be in moderate community-acquired pneumonia patients. The moderate indication, we actually prefer will have more benefit in the severe population, but the moderate population was requested and highly recommended by the FDA for the start. So we decided not to argue and start and collect safety there. This will also be a single ascending dose. I'll do a shameless plug. We have 4 sites right now that we've got 2 started, 2 in the various stages have started. If you have a site in the US that's interested, please reach out to me. I have no problem with the shameless plug. My CEO is giving me a lot of grief to get this enrolled fast. So any help would be appreciated. In addition to that, we're also starting an adaptive phase 1B/2A in cystic fibrosis patients. We're going to start with a small safety escalation study. It's going to be unblinded as this is a rare population. We already have healthy volunteer data. We're going to just make sure that it's still safe in cystic fibrosis patient, and then we're going to go into a dose finding study with an interim analysis that is going to have adaptive design to basically indicate continue as is, increase a dose, decrease a dose, remove a dose, or stop the study is futile. As I mentioned, there is lots of broad indications. We have several listed here. This is, I think Tobias had mentioned this earlier, investors sometimes dictate what indications you go after. These are a lot of the indications that we have found have been attractive to the investors, more of the lower respiratory indications, such as pneumonia, or an outpatient, such as cystic fibrosis. We also recognize that COPD would be a large field that could have benefit greatly here. We're a 6-person company. Hopefully by then someone else has come along to help us move this product along. We also recognize bone and joint infection has a lot of probability here as well. I didn't mention, and I won't get into too much detail, this is just in lead optimization phase, but we are developing a vaccine using the same antigen that we developed CMTX 101 as a preventative measure against bacterial biofilm. We call this CMTX 301. And this is the graphic up here is actually out of our scientific founders lab where they did a viral infection, kind of my kid has a cold, and then 2 weeks later they get an ear infection concept model. So they were able to actually prevent the ear infection in otitis media model. We are looking at it as an otitis media model vaccine, but we're also exploring other things as preventative mechanisms or even a therapeutic vaccine. So take a deep breath here. We have a game-changing novel approach to addressing bacterial biofilms. We are pathogen agnostic. We are antibiotic agnostic. We do have the ability to potentially even address AMR concerns out there as we are restoring sensitivity to frontline antibiotics. We have a pipeline of CMTX 101 and 301. We have a fairly well-established team of experts that are helping us develop and influencing our decisions. And we have had support from CARB-X, approximately 9 million support from CARB-X. And we also have an IDCB grant out of the NIH for approximately 3 million. I can open up the floor for discussion.
Very good, thank you. Then we are discussing new modality, and this is definitely a new modality. This is open for discussion. Yes.
Without modifications, it's got a half-life of about 10 days, which is great for the acute infection. We're not seeing any anti-drug antibodies right now from our healthy volunteers, so you could potentially dose again for chronic. But that is something we could explore to modify the FC to give a longer half-life. It's also from the therapeutic. We speculate you might be able to even do that with the vaccine for more of a chronic condition.
Yes.
We are trying to start treatment within 24 hours of their first administration of antibiotics. We really want we're right now a single dose treatment and a single dose.
Does it cross the blood-brain barrier right away?
No, no, it's an antibody. It doesn't have the ability to cross.
Thank you for this new treatment. Concerning patients with bronchiectasis or cystic fibrosis, as you said, how many days and what dosage do you use or do you advise?
For
bronchiectasis and with Pseudomonas infection, with chronic Pseudomonas infection?
That's a good question. We would have to look at more of the chronic condition right now, similar to the CF patients, which hopefully will help inform also for the NCFB patients, that if it's going to be chronic, multiple treatments, is it going to clear? We're actually exploring that, some of the questions with our initial CF.
Yes, because concerning biofilms and Pseudomonas, and
Yeah.
I saw that there was a good response in vitro and in vivo with Pseudomonas in your
Yes.
slide. One of the targets in our population are chronic Pseudomonas infection. And it means COPD patients and bronchiectasis and cystic fibrosis.
Yeah.
And it's very challenging.
It is, yeah. Yes.
Did you study it in bacteria without biofilm? There's any antibacterial direct activity of the compound? Because I didn't get it really. Biofilm is really effective. They are infectious infections. I mean,
Yeah.
The idea, the prevention idea, and so on. But in acute bacterial pneumonia, there might be some direct antibacterial.
There's no direct antibacterial effect. Its target, it's actually very specific to its target, which is the DNAB2, which does exist internally in the bacteria or in the biofilm, but it's not expressed on like the outside cell wall of the bacteria. So it doesn't have the bacteria themselves if they're not in the biofilm, all of the target is internalized.
Then the point is that when the patient arrived to the hospital with 3 days incubation period of a pneumococcal pneumonia, how much of the biofilm is there? Well, there's some data indicating that probably is a lot. But this will be, I remember discussing with Veronica, this is a paradigm change of what we thought that for this acute pneumonia, there's no biofilm. Yeah. When you go into the chronic pneumonia, you need to have something chronic to have the biofilm. But based on some animal studies and based on the results, seems to be that by the time that it's 3 days of pneumococcus, that the pneumococcus probably as may happen in the laboratory. You get a Staph aureus, they develop biofilm immediately. They want to go into biofilm. We give the options to develop biofilm, but it may be that once the bacteria is in the alveoli, they immediately, as they multiply, they form biofilm. If this is the case, then the study will work. If this is not the case, yes, but it's, and the idea is then if really we have pneumococcal in biofilm from the very beginning, on day 1, and we give antibiotics that really destroying the biofilm, increase the susceptibility of all the bacteria, and then giving antibiotics, everything is simple. It's a lot of possibilities.
This is kind of a comment because our hospital looked at being a part of your study, and the problem was that the pneumonia patients are ward patients, and we didn't have the capability to do all the blood draws. If you had allowed severe patients who have arterial lines and all that are in the ICU, we could have participated.
We'll keep in touch for the phase 2. Julio was part of this. The FDA would not budge. We got them to budge from what we called mild outpatients to go to, we thought, moderate in the middle, and we had to come up with a huge definition for moderate for them as well, but we could not. We thought as a small company taking on the FDA would be very difficult.
And the FDA is also very, very concerned not to go into serious patients.
Yeah.
No, really, I mean, this idea that all acute infections are biofilm is something that has not been, yes, it's a new way, it's a new approach. Good. Any other comments or questions regarding this topic? Okay, thank you, Veronica. Excellent. Thank you very much.